Good afternoon. So my name is Stephan REILING. I'm a scientist with the Novartis Institutes for Biomedical Research, which is the research arm of Novartis, which is a large pharmaceutical company. And we only do research, and what we're researching is to identify the next generation of medicines to cure disease. And identifying medicines, biomedical research, really has become just this big data munching exercise where you generate lots of data, you analyze even more data, and all of this at the very end then gets distilled into a small pill or injection or in some treatment. So what I want to talk about today is a project that we've been working on for almost 3 years now, and we're now getting the first results, and they look very promising. So what I'm going to talk about is how we have combined lots of heterogeneous data and integrated that into one big graph that we're now using for querying. Some of the data that we're putting into this graph is coming from text mining, and we're doing this a little bit different in terms of how what we extract from the text and how we do the pattern detection there. I'm going to talk a little bit about that, and then I'm also going to have examples of how this then actually can be used. But before I go and start with this story, I need to describe why we're doing this in the first place. What is the situation that we're trying to address? And this, okay, so looks a little bit better here. So it's about images or capturing complex biology. So in the past, when you wanted to understand biology, you would be interested in a protein, you would isolate the protein, you would purify it, you would put it into a little test tube, and then you would characterize it. And if you wanted to identify a compound or something that acts on it, you would put the compound into the same test tube, and you would see what it does to the activity of that enzyme or some other protein. So that is very reductionist, but it allows you to get very precise measurements if you want to. And we have decades worth of data along those lines. That was, for a long time, was the workhorse for drug discovery. And so you identify from this a drug or an initial compound that goes into a multi-year effort that at the very end then results into a drug that comes onto the market. And as we found out, if you do this, biology cannot be reduced to a single protein in a test tube. It's much more complex. So over the last couple of years, probably the last 5-8 years or so, all the pharmaceutical companies and also research institutes across the world, they are now starting to try to capture the complexity of this biology. And one way to do is to run these high content screens. So this is a snapshot of a cell culture, and the actual image is much larger. I took a crop of it. And we run large-scale screening assays on these kind of things. We have these big automated microscopes with lab automation, and we can easily generate terabytes and terabytes of images along those lines. So what you see here in white is basically the cell body. The red circles are the nucleus, and green are the nuclei of cells that do not contain a certain protein. And so let's say the goal that you have is to increase the number of the cells that express this protein, which is here colored white. So you add some treatment to it. It could be a compound, it could be a small RNA, it could be anything. And then you see that not only do the numbers change, but I hope you can see that there is more going on. The shape of the cells is changing. Now you add a different compound to it, and something else happens. So we call this a phenotype. And we're now running very large-scale screening efforts where we use not a numerical readout, but we're trying to capture these phenotypes, and we try to use these phenotypes for one, to understand the underlying biology, and also to identify in a better way the compounds that will go into this long process to get at the end a drug. And this is only getting worse. I mean, there is now, lab automation is progressing. We can generate more and more of this data. There is now movies coming up where we take live cell imaging and we follow the cells over time. There is 3D, we take 3D slices and so on. So there is more and more of this data coming our way. So here is the basic problem statement. So what we have these decades worth of this reductionist data. We have these large compound libraries, they're annotated, we know what they did in the past, but now things are changing and we're getting more and more of these imaging data and these phenotypic assays. When we try to analyze this, it becomes much more apparent that we need to have a way to scale biological knowledge, or to have a system that allows us to store biological knowledge and then run queries against it, and so that we can use this to analyze this data. And I still don't know exactly what biological knowledge means. So, but we're going to make a system for it. So one of the sources of information, sorry, I should mention this. So this at the bottom is the kind of the logo of the department that I'm working in. And so it's this triangle where you have at the top, you have a compound, you have gene and a phenotype at the bottom. And the way we think about it is that for a successful drug discovery, you need to be able to navigate this triangle. And from the data that we accumulated historically, we are very strong on this one edge of the triangle between the compound and the gene, but not so much on the other edge. So we're trying to fill this knowledge gap. So I mentioned that one of the sources of information that we're putting in there is text mining. And so here is some scientific text. And there are tools available that will then identify things of interest, entities of interest in this text. And so here we're identifying compounds, genes, and also diseases and processes and so on. And some of these tools, they cheat a little bit in that such they tell you only this is a compound, but they don't tell you what this compound is. So one of the things that we have been working on to identify exactly what the compound or what gene it really is, and we then re-engineer basically the chemical structure from what we're identifying in the text. And we also do things where we're trying to identify relationships between the entities in the text and come up with statements, the compound inhibits this target, and so on. For the talk here, the corpus that we had been using initially is PubMed. So there was already a talk this morning that mentioned it. So it's the National Library of Medicine. It's an incredible resource. They have been, since 1946, they have been collecting articles of about 5,600 scientific journals, and they make the abstracts of these articles freely available. It's a really incredible resource. What is sometimes not appreciated is that when an article or an abstract gets entered into this library, it gets tagged by human experts. And they call these mesh terms, the medical subject headings. And so for the text that is on the left, on the right here, I'm just giving you an impression how many tags you actually get. So, and the nice thing about these tags is I will show that they're organized and they're coming from human experts, and sometimes they describe the article in a way that you would not be able to tell from the text, because there is this human curation that basically is happening. So I talked about that this is organized, and here I'm just showing a little bit about how this is organized, these mesh terms. So they're organized in these trees, and here I'm showing 5 of these, the ones that we're interested in. In total, there are 16 trees, and so they also have things like geography or occupation, which for our purposes is not of that much interest. And there is about 250,000 of these tags that the NLM is using. And I have to make one disclosure here, is that in reality, they don't look quite that nice. So the overall organization is a tree. There are some connections between the branches, and so the actual layout of this doesn't quite look that nice, but in general, it's organized as a tree. And I'm just trying to show you a little bit how deep and how rich this is. So this is sized by the number of articles that are annotated with a specific tag. And you cannot see the text. So here's, I'm just zooming further in. And so here you can already, just from the tag, you can tell that the article is going to cover a specific subtype of a receptor. So that's already very rich and detailed information. And we're using this. So the disadvantage of this is that from the text mining, we're getting entities, we're getting relationship between these entities. Here we can only tell the tag is in the abstract, or it's annotated as this, and we would like to combine these and use this and put this into a graph. So how do we do this? We're exploiting the fact that we're looking across all of these 25 million abstracts. And when you do this, and so here's a little toy example where I have these 4 articles, and if you do this and you look across these articles, you will see that some of these entities, like compound A and gene 1, occur together quite frequently. So we're using association rule mining to establish what the association or the probability of co-occurrence for these entities is. So we're not using the verbs of the sentence, we're only using these entities. And so we calculate the lift, it's also known as the lift, and we scale it from 0 to 1. And then if it's above a certain threshold, we then say there is a relationship between, in this example, compound A and gene 1, and that is a relationship between these nodes that we put into this graph. So here's a way that from the text, we can generate, based on this statistical analysis, relationships in a graph. So what can you do with this? So I already mentioned that we have this uncertainty. So we're scaling it from 0 to 1, and we call it an uncertainty. The reason for that is that later on, we're going to do a lot of things where we do graph traversal. And we're going to use this as a distance measure in the graph. So the association rule gives us a confidence that they are associated, and we just take the inverse. A high association has a low uncertainty, and that's the way that we're entering this into the graph. So on the first slide, you saw we have this little triangle that we're trying to navigate. So now I can just do a triangle query where I say, I want to find a triangle between a compound, a gene, and a phenotype or a disease. And I want to take the sum of these distances of these 3 edges. I want to use that to rank these triangles. And I want to find the one that has the lowest uncertainty at the top. So when I do this, one of the first triangles that comes up is this triangle that has tafamidis, amyloid neuropathies as a disease, and transthyretin as an enzyme. So what gives? We found this. How can we validate if this is knowledge, common knowledge, or not? So we go to Wikipedia and we check. So this is taken from the Wikipedia page about tafamidis, and this is one of the first sentences on the Wikipedia page states that tafamidis is the drug that is used to treat these amyloid polyneuropathies, and it's caused by a transthyretin-related hereditary amyloidosis. So this is the validation, or one way of validating this, that with this approach, with this association, we capture this. We can do this again, or the second one that comes up is another triangle, and this is now also on the Wikipedia page. It talks about Canavan disease, and in this case, the compound is not the drug treatment, but it's actually the accumulation of this compound that causes the disease. The point I'm trying to make here is that we are losing more details about this relationship. In the one case, the compound played the role of the drug. In this case, the compound plays the role as the causative agent, but we get this overall picture, and that's what we care about. So we go to Wikipedia, we check, why wouldn't we just load Wikipedia? So, yeah, you cannot see much there, but so this is the Wikipedia page for this compound that was on the second triangle on the right. So this is N-acetyl aspartic acid, and as far as Wikipedia pages go, it's a rather short page. And really the only thing that is on this page is this relationship of this compound with Canavan's disease and the enzyme on the previous page. So what I can now do is I can go into this graph and I can say, now for this compound, rank order me the diseases that are associated with it. And when I do this, these are the top 5 diseases. And the first one is Canavan's disease that I showed before. And then there is 5 more. I'm just getting here the first 5. And since this is all coming from the literature, from PubMed, I can now go and say, well, what is the supporting evidence for this association? And so here is then one article that you can pull up, and directly in the title it says that this compound plays a role in Pelizaeus-Merzbacher disease, which I didn't know. Okay. For the analysis of these cellular assays, we are actually not that much interested in diseases. So I can basically take the same query that I had before, and I just exchange disease for cellular component, for example, and I get the results on the left. And if you remember, this Canavan's disease was a neuronal disease, so that the components that I see here are all related to central nervous system. On the right, I'm doing the same thing. I just replace now cellular component with biological process, and here I'm getting the list of biological processes that are associated with this compound. And for the analysis of the cellular assays, these cellular processes, that's really something that we're interested in. So by exploiting this, and all of this is coming from these mesh terms. So this gives us a very broad annotation or knowledge of what is in PubMed. So this was the text mining I was talking about, that we're doing this integration of heterogeneous data sets. And so here's I'm just showing what we have put into this graph database so far. So the green ones, the top 3, I covered on the previous slides about the text mining. There is 10 more. The selection was done such a way that they should complement each other. So we have toxicogenomics database in there. We have protein-protein interactions. We have systems biology and pathways, proteins and gene annotations. All of this we integrate, we match it all up in terms of the identifiers and the objects that are in there. And we're getting about 30 million nodes that we're now putting into this database. And we're also putting the articles in there. And the majority of these nodes are these articles. For us, what is especially important is we have about 2 million compounds. So that for us is a really important number. But we're not going to use this to identify new genes or anything like that. For us, this is all about relationships. That's what this connecting the dots is. So we have about 91 different relationships, and so a total is about 400 million relationships. And the 91 different relationships, that's just the richness of the biology and the underlying data that we're getting. So I'm just showing briefly what these relationships look like. So we have anything from a protein phosphorylates this protein, the compound affects the expression of this protein. So we're trying to be pretty broad here. And just a little bit about the technical side, how we're doing this. So we have this infrastructure in place where the PubMed articles, the abstracts, first go into the Mongo database. And that is really what is driving the text analysis. The PostgreSQL database that is there in the middle, the main reason is that we are using existing data warehouses at Novartis where there is already work that has been done to do some pre-summarization of data, internal and external, and we can just do ETLs to get this into this Postgres database. And that is, for us, very fortunate. I mean, there's many years that went into these upstream data warehouses, as you can imagine. The way we're using Neo4j, or how we getting data into Neo4j, is we go actually through the CSV batch importer. At this point, we're still figuring out exactly how we do the text mining and so on. So doing this staging through the CSV files, it has worked very well for us. So, okay, we can find things that we found on Wikipedia. What do we really use this for? And so here is one example. So I talked about that we're running these imaging assays, and in a lot of cases, we know what we're looking for. We want to see this phenotype. We don't want to see other phenotypes. So we can categorize, if we test compounds, for example, we can categorize the compounds into active and inactive. And if we now want to analyze this data, what we had done in the past, and we can do this now also here just in a graph form, we can look for what are the targets what are the target annotations that we have for these compounds. And we call this target enrichment. We have been doing this based on relational databases, and it's a hit or miss. But what we can do now is we can now go into the graph and run queries where we are saying, what are all the nodes that we can reach for each active or each inactive compound that is within a certain distance? And this is where this uncertainty comes back. And the idea is that we're trying to identify nodes that are in there that can be reached from the actives, but not from the inactives, or are much closer to the actives than they are to the inactives. And the idea being is that this is something that the actives have in common, and it could be a gene, it could be a biological process, it could be a pathway that really differentiates the actives from the inactives. So just a little bit the technicality here. So we run these queries, we get rows, every row says compound, the node, and the distance, and then we pivot this into a distance matrix where every row is a compound. We have an indicator column. We have here the distance to these nodes, and if it cannot be reached, we just set it to a really high value. And I talked about how we're doing this for hundreds of thousands of compounds. This was an example that was given to me by a biologist, and it has 34 compounds. So that makes it easy to display here. So we then run a partitioning. We're trying to identify a cut that separates in this distance matrix, that separates the actives from the inactives. And the example here, we can find a cut that splits 12 of the 14 actives on one side and all of the inactives on the other side. And the way that these decision trees or partitioning methods work, they will also find other cuts that work as well. So this is called a surrogate split, and we also use those. And so in the example here, we found 2 of these internal nodes that we're looking for. And this is what this looks like. So these are now just the active compounds, and the blue are the compounds, the red are the nodes that are in this network, and in this case, these are all genes. And in yellow highlighted are these 2 central nodes that seem to determine if the compound shows the desired phenotype or not. And in the example here, this turned out to be true. So the other reason why I'm showing this is that the relationships are colored by the data source. So green, and these are all the connections that go from the compound to this initial layer of nodes. Green means this is our internal data. The gray, which is all the rest, this is coming from these association rules. So this is really something where we wouldn't be able to find this without integrating all of these data sources and also doing this analysis from the text mining. So, but is that really true? So in this case, we wanted to find out these association rules, what is going on, why couldn't we find this differently? And so this is this again. And I'm not just trying to find the connection in this graph by ignoring the association rules. So every relationship is annotated with the data source that is coming from. So I can run the same query by eliminating the text mining. And on the left side, you see this blue box. So on the right, this is only these 6 nodes that are shown, and the ones with the yellow circles are the ones that are in this blue box on the left, just to keep this a little simple. And so you can find a connection between them, but the connection is looking much more complicated. And if you sum up these uncertainties, it's a much weaker statement. So the role that these association rules results play is that they provide shortcuts basically across all of these underlying data sources. You can now take these as a shortcut to identify something, and then you can use this to drill down to better understand what is going on. And this is where these abstracts come in, because most of this is coming from the literature, even though it's coming from a systems biology database. So if you have these association rules, you would then also like to know what is the support for this. I talked about this before. And so this is the same query that I ran before, and all of these purple spheres are the articles that constitute the evidence for this association. And since we have them in the database, you can click on them and you'll see what there is. And the one that is shown there, it talks about that. It talks about the same relationship that we identified, and it also talks about a compound that should behave similarly to the ones that we tested. So not only did we find a hypothesis in this graph, but we also now have a way to test the hypothesis and see if it behaves in the way that we would predict from this. And we have not done that yet. So also almost coming to the end, and wanted to do a little bit of reality check and circle back. So this is the result that I showed before. And the very first time we got this to work, it was not this, it was something that was similar. We also were trying to identify what is the connection between things. So the very first time we did this, we took the results and we went to the biologist and we said, what do you think? Does that make sense? And the feedback that we got was, I knew that. There was a little bit of disappointment maybe in there, because the biologist wants us to find these really novel insights he didn't know about, and it's all cool and earth-shattering and so on. So that's not how this is going to work in most cases. I mean, you get novel insight into how your compounds behave and why they do what they do, but it's not always going to be this smash hit of earth-shattering results. But I talked on one of the first slides, I talked about this, we're trying to capture this biological knowledge, and that this is very fuzzy and I don't know exactly what it means. But here we have something that is non-trivial to deduce from the data, and it's mostly about relations between biological entities. And if you now take this and you go to a biologist, and the biologist says, I know that is biological knowledge. I mean, that's kind of what we're trying to capture here by combining the literature and these data sources. So where is this going? So I just have here a list of things in the short term that we're trying to do with this. And some of the things I glanced over a little bit. So we're trying to do a better job with the text mining. We have now started doing full text analysis of full text, not just abstracts, mostly for purposes for data mining internal documents. And also this concept of this uncertainty, it was kind of like, yeah, we just take this threshold, and there's a couple of things where we should be a little bit more rational about. What we don't have at this point is an automatic update. I mean, every day there is new articles that get published and get put into the library. So we don't have an automatic process that is doing this. And there's always more data. So What is not in there on purpose is a lot of the genomics data, genes, gene expression data, and so on. So once we put that in there, it's going to at least double the size of the database that we have. So I also would like really to acknowledge the people that directly or indirectly contributed to this project, to this effort. So I'm in this ISLD group on the left there, and it was really great collaboration there. I already mentioned the importance of the text mining, so that's why the text mining group is up there. And under DMP, I want to give special thanks to the 3 guys that you see there, because those were the ones that were initially responsible for creating these data warehouses that are already summarizing a lot of this data. And then on the right, I really have to thank all these incredible public resources and public websites where you can download all of these databases. That's really the only way that we can make this work. And I thank you. And if you have any questions, I'm more than happy to answer them. Thank you. So the question was, when we find these relationships, how much manual curation is there? And the answer is none. So we tweaked parameters and we tweaked the text mining, but the goal is to have an automatic process where we can feed text in there and we take the results. And some of the statistics we use to validate and to tweak the parameters of the text mining. Yes.
So any hypotheses in terms of the kind of tools you might be
So we started, so the question was that I talked about we're going to use verbs for extracting this and so on. And we started doing this. So if you go and you do information extraction, open information extraction, a lot of these tools, what they do is they identify subject, verb, object. And that we are doing, but from what we can tell, we're missing a lot. Yep. So it's Forrest Gump, as an example, I like chocolate. If Forrest Gump had a PhD, he would say an individual was exposed to several different food item groups. Statistically significant preference for chocolate was observed. So you end up with these much more complicated sentences, and the simple subject, verb, object, you lose. Yep. So, and this is something that we're working on right now.
So do you think classification or hierarchy terms like in terms of compound and also diseases, there are taxonomy?
Yes.
Taxonomy to organize or classify them. Do you take those into consideration when you organize, when you do text mining?
So the diseases and compounds are organized in the mesh. So that's what we use as a taxonomy, or it's not really an ontology, but there is this organization of diseases and compounds in mesh. That's what we're using. So there are other ontologies and taxonomies, and initially, so last year, we tried to use multiple ontologies with it, and we got into a little bit of like an analysis paralysis. Just there was Medra and so on, and so we said, well, let's first see that we can get this going, and defer this a little bit. So we first wanted to see, does this actually work? Can we now show impact on the projects that we're supporting in terms of drug discovery? And I'm pretty sure we're going to come back to these ontologies, but for now we deferred this a little bit.
How do you handle dirty data? For example, some research articles, and they are
Yes.
cited or they are considered as fake data.
Yes.
Nobody can reproduce it.
Yes.
So how do you clean out all those dirty data?
So there is the data reproducibility. There was there's this ongoing discussions about that you have these articles and then they turn out that what they said in the article is not correct. So that's also the purpose of these association rules. So we're not looking for something that is in a single article. So in association rules, you have these two parameters. One is how strong is this association and how many articles, in this case, support this. So you have a minimum support. So if it just occurs in one article, we don't use it. It has to be, I believe right now we use 10 articles or so where this has to be in. So that's a little bit of protection that we think we have in there, but it is true, especially if you have things like after 10 years where this was accepted dogma, you have this thing and now it turns out all of this was false. There is nothing much we can do that would save us from that. But this is also the purpose a little bit of the association rules. Yes?
How long did it take you to kind of develop this methodology and kind of start from
Yeah.
where you had a vision for this and then so I could work with it?
So this all sounds very smooth. It almost looked like, it was like, woo! It wasn't. So there was a little bit of twists and turns and so on. So the way you should think about it, it's just one big bootstrapping operation. You start from nothing and you say, how can I go about this text mining? So you look at tools and then you say, how can I validate these tools? And that is really why we initially used these mesh terms. Because if you have a tool that tells you, I found this compound in this article, then this should match up with the human annotation that is in these mesh terms. Yep. And we didn't initially use Neo4j for doing this. We used the triple store. And that's really why we got into this whole discussion about ontologies and OWL and RDF plus and all this fun stuff. And so we have been using, so we had the underlying concept in place for quite a while. We got bogged down. And then beginning of the year, I basically said, well, let's get something done that is much more practical. And that's really that's the result of this. So we had initially a much bigger vision for it. And this is actually a little bit of a scaled down version for this. I only talked about like the short-term goals. This could go much further than just what I showed here. Also in terms of the domains that we're including.
So kind of part two, that would be, okay, so how is Novartis using this to define or to identify what drugs or what niches it wants to go after? How does that work?
Yes.
User access?
Yes. So the group that I'm in, we are basically the data scientists, the data miners, in a department whose goal it is to identify targets and to identify compounds that go into the lead optimization project. With this phenotypic assay, the nice thing and the bad thing about it is that you can run assays that you don't really understand. And you run these assays, you take these images so you can understand it. So this has become a much more circular process where we go through these iterations of taking these images. You should think about it like you find a new continent and you're trying to make a map. And you first make these high-scale images from a satellite, and once you have figured out where the cities are and so on, you start zooming in. And that's what this is also about. So the goal is not just to identify compounds, but to figure out if these cells turn green, what causes them to turn green? Is this something, and we observe this in this disease, if you can figure out why this happened, and if you can figure out the compounds that make this not happen, then we can go and that becomes the next drugs, the next drug. Did I answer your question? Yes.
So how do you handle side effects for the multi-compound versus multi-disease?
With that, right now, that's not in there. But it's one.
You have a single compound, single disease triangle, right?
Yes.
So we cannot handle like or seeing that multiple compound with different dosage that will have different side effects
So, so right now, we're looking at single assays. Now we store all of this information. And once we have this method going better, yep, as I said, there is all kinds of plans, all kinds of directions where you can take this.
So you said initially you're using some other thing instead of Neo4j, and you're now using Neo4j.
Yes.
Why you use?
So the question is, why did we switch to Neo4j? I think I opened a little bit of a can of worms there. So before we use a triple store, so difference between Neo4j and a triple store is that Neo4j has nodes, and the nodes have parameters, and then you have relationships. A triple store doesn't know about nodes, it doesn't know about relationships, it only knows about triples. Yep. So let's say you have a compound, and the compound has a molecular weight. In the triple store, this is a triple. And if you now have a relationship between a compound and a target, what we are interested in, this is also a triple. Yep, so it's all the same thing. I mean, the triple stores are super flexible, and you can definitely do things with the triple store you would have a hard time doing with Neo4j. Don't quote me on this. I'm not quite sure. I might be proven wrong. But all of what we're doing with this is about graph traversal. So we're trying to find a connection between here and here. And when you try to do this in a triple store, you always have to tell the triple store, oh, this is not a triple that you should consider, and not this type of triple, and so on. Because there is no distinction between what in Neo4j is a property and what is an actual relationship that you want to use for your graph traversal. Yep. And so the other thing is that a lot of this can be cleaned up and avoided by using the proper ontologies. And that's where we got bogged down in this discussion about ontologies. We had a list of 25 ontologies or something like this, and they didn't really match up, and it was, we probably have to go back to this and revisit this, but for now, we're using Neo4j. Yes?
So do you have any idea or any thoughts about how you might incorporate ontologies in the future?
So there is so I know that there were, I think there's also a talk here about how you can have something like an ontology in Neo4j. I'm not sure that we want to have an ontology in Neo4j. So in Neo4j, this would be much more like the concept of the schema and the concept of constraints. So I have not used Neo4j long enough. So we're only using Neo4j since the beginning of the year. Yep. So I have not used it long enough to really see, to really know if we need ontologies in this or not. There definitely has to be a map. A lot of this data is published as RDFs with an ontology. There probably is definitely a mapping step necessary. But if we can get by with schemas and constraints in Neo4j, I'm not sure that we want to introduce ontologies. Yes. Oh, so we're done. that was that. Is there a last question, maybe? Something urgent? So, oh, okay. Sorry, can you repeat the question? Yes. So the question is, give an example of gene expression or gene data that we would like to introduce. Yes. So one of the things that we're really keen on, so we run these cellular assays, and we're using different cell cultures, different cell types. They behave differently. So we want to know why they behave differently. The difference between these cell types can be described by looking at their genome or by differences in the gene expression. Does that answer your question? Let's discuss afterwards. We're running out of time. So I mean, we can discuss offline. Thank you.